So the cytoskeleton is the structural framework of the cell. And similar to our bone skeleton, the skeleton serves as to maintain the cell shape and internal organization of the cell. It's found in all eukaryotic cells and allows for organelle and vesicle movement within cells. It also allows for cell motility in some cases, so similar to our bone skeleton. The framework of the cytoskeleton is made up of actin filaments that are typically called microfilaments, intermediate filaments, and microtubules. Actin filaments or microfilaments are the thinnest cytoskeleton proteins. They are made of semi-flexible strands of actin. There is a dense cross-linked network of actin attached to the plasma membrane that strengthens the cell surface and helps to resist cell compression. The actin filaments may also be involved in cell motility or changes in the cell shape through the formation of pseudopodia. And it's involved in assisting materials as they enter and exit the cell in the processes of endocytosis or exocytosis, respectively, which we'll talk about in the next chapter. Intermediate filaments are intermediate in size between the microfilaments and the microtubules. They are made of tough, insoluble, rope-like strong structures that are a tetramer of 4 fibropolypeptides twisted together. They provide the support and help cells to resist pulling forces. These filaments attach to the desmosomes, which link individual cells together. So the intermediate filaments bind to these desmosome plaques from the internal side of the cell and present compression or separation of the cells due to compression on the cell layer. These same intermediate filaments are called neurofilaments when found in nerve cells, or keratin filaments when found in skin cells. Microtubules are the largest of the cytoskeleton elements. They are made of hollow tubes of tubulin subunits. So they're composed of hollow tubes that are made of tubulin subunits. These tubes are constantly being assembled and disassembled. At one end they're being disassembled, at the other end they're being assembled. Most radiate out from the centrosomes, or that include the centrioles. Their main functions are that they may be also help with microfilaments and determining the overall shape of a cell. And they also keep organelles in place by being tethered to microtubules, because the microtubules are tethered, because the organelles are tethered to the microtubules, which holds them in place within the cell. And substances can use these microtubules as motor proteins, with motor proteins that use microtubules as tracks. Also, microtubules are what make up the mitotic spindle, to talk about in several chapters from now, but we've touched on in lab before. The mitotic spindle that is involved in separating the chromosomes during cell division. So an overall review of the cytoskeleton components are microfilaments, which are strands of the spherical subunits actin, and intermediate filaments, which are a tough insoluble tetramer of 4 fiber rope-like proteins. And microtubules that are the hollow tubes of spherical protein subunits called tubulin. Microvilli, flagella, and cilia. Many cells have surface extensions called microvilli, flagella, or cilia that aid in absorption, movement, and sensory processes within the cells. Like we talked about before, there's only one type of animal cell that contains flagella, and that's the sperm. These are microvilli, flagella, and cilia. Microvilli are extensions of the plasma membrane that increase the surface area of the cell. They are anchored by actin filaments. These actin filaments, you don't see it here, but they move up into the villi. And they do not contain what are called axonemes. Microtubules in flagella and cilia are arranged in an axoneme, which is that has a 9+2 microtubule array. 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2. So there are 2 central microtubules that consist of 13 protofilaments arranged in a circular tube. Each of the 9 outer microtubules also consist of a pair of microtubules that share a common wall. So 13 protofilaments, and they're a doublet. The flagellum is a whip-like structure that allows for locomotion of the cell. They are found in all 3 domains of the living world: bacteria, archaea, and eukaryota. This is sperm, and this is the Helicobacter pylori. If you know somebody or you've dealt with stomach ulcers, this is often your culprit for that. The hair-like appearance of flagella or cilia can be misleading since the entire structure lies within the plasma membrane of the cell. The plasma membrane here just extends to cover the flagella or the cilia. A cross-section through this shows the same structure that we were looking at before, the axoneme, that is made up of microtubules. So remember, you've got that 9+2 arrangement, and each microtubule is made of 13 protofilaments, and the doublets on the outside share a wall. Compared up against this structure, they're identical. If you compare it up to the basal body of the centriole, they're identical. In that the centrioles and the basal body of the flagella contain 9 triplet groups of microtubules. Within the cell, if the pair of centrioles are found next to each other, they are usually at right angles. What you're seeing here is a cross section through one. And a cross section through the other one, and indicating that they're at right angles to one another, or perpendicular to each other. Centrioles work in pairs with each. During cell division, centrioles work in pairs with each moving toward the poles of the nucleus and the cell when it is time for cell division. Cilia, similar to flagella but much shorter, not as long, alternate in their movement between a power stroke and a recovery stroke. This results, so they fold down and then they flip back up. So fold down, power stroke, then they flip back up. This alternation produces a current at the cell surface that moves substances along the surface in a forward direction. If you've ever dealt with a cold, and you frequently will cough up some mucus into your lungs, and then you work to swallow it. And when it's swallowed, it goes down into your stomach where it's destroyed. You have cilia along your esophagus and your bronchi that allow you to cough that up and then swallow it. An overview of the organelles. So a nucleus is present in both animal and plant cells, and it directs cellular activity and stores hereditary information. The cytoskeleton, which is present in both types of cells, provides structural shape and support and enables cellular movement. The mitochondria harvest energy for cellular functions, and they're present in both types of cells. Lysosomes, present in both types of cells, digest and recycle cellular waste products and consume material. The rough ER, present in both types of cells, modifies proteins that will be shipped elsewhere in the organism. Smooth ER synthesizes lipids and detoxifies molecules. The Golgi apparatus processes and packages proteins, lipids, and other molecules. It's also present in both types of cells. The cell wall is found in plant cells and in prokaryotic cells, but not found in animal cells. And it provides structural strength protection, and increased resistance to water loss. In animal cells, vacuoles are sometimes found, but they are found in all plant cells, and they work to store nutrients, degrade waste products, provide pigments, and provide structural support. And the chloroplasts perform photosynthesis, and they're found only in plant cells and some prokaryotic cells. Brief overview here. So a compound light microscope focuses light rays that pass through a specimen to produce an image. Transmission electron microscope uses a set of electromagnetic lenses to focus a beam of electrons that pass through a specimen to produce an image on a fluorescent screen or photographic film. A scanning electron microscope aims a focused beam of electrons onto a specimen that's been coated with a thin metal layer. That beam of electron excites some secondary electrons in the metal that are emitted from the metal, and their emission pattern is used to create a three-dimensional image of the specimen.